Okay, so the link for that next lecture is right there. And like I said, we'll be doing a lab next class kind of together on Wednesday. One thing I want to point out while I'm here, and you guys can see my screen here, this is this extension right here. It's called Paint on Tab. Do you guys have that extension? Okay, well, let's get it real quick. Okay, so if you would just, you can open up a tab and we're just going to search. We'll just do Paint on Tab tool. Paint on Tab tool. And if you then click on kind of the first thing that pops up there after your search, do you guys have where you can add it? Yay! They granted you permission. I asked them to give you permission. Let's go ahead and add that extension because it becomes really handy in your breakout rooms. So again, it's called Paint on Tab tool. And you should be able to click on the first thing that pops up, which is right there. Click on that. It's going to take you to the store, and then you can add your extension. Okay. All right, so one thing when you guys are in your breakout rooms, you guys are going to be working together to work on this assignment. We're going to do some parts together to review what you need to know from lecture 3.1. One thing that is kind of helpful, if you want to use this feature to draw on there, you want to open up to make sure it's like in some kind of tab. So you'll want the assignment open in a tab. Hopefully it's going to open it for me here. And then when you click on that extension, this will pop up. And so if you guys need to draw or need to, someone doesn't know like where you're at, whoever's presenting can write and stuff like that so that you guys can kind of work on these parts together. This is just a handy tool that can help you do that. Okay. All right. Okay. So with my document cam here, let's take a look at that worksheet 3.1. And I'm going to go down here to number 2, right? So here I have 5 main types of organisms on this planet. This covers everything, okay? Everything on this planet can either be classified or categorized into one of these categories. There's no outside category for anything on the planet, okay? That's a living thing. And I know that some of you are like, Ms. Spencer, why do we have to know all of these details about all these, it seems, insignificant organisms, right? And honestly, the reason why is kind of dark, to be honest. We want to learn as much as we can about other organisms. So one, we can kill them when we want to kill them, so that we can manipulate them and use them for our own good. Like I told you, it's kind of dark. Like I feel like humans are the bad guy on every single cartoon that you guys have watched, right? There's always a bad guy who's after something, and like, you know, the super pups or whatever have to, you know, fight off the bad guy. And the bad guy is trying to manipulate things, destroy things, right? And honestly, that's just the main reason why we have to know a lot about other organisms, is because we want to be able to use them or we want to be able to destroy them, okay? So here's an example where we need to destroy an organism, and we don't want to destroy your cells in the process. So this girl went blind because her contacts were infected with amoebas. She didn't change them. Now, amoebas are a type of protist. They're a single cell organism. And there's different types of amoebas, and they can cause different infections in different areas of your body. And so she went blind because of the amoebas that were destroying her tissue in her eye. And so, again, this is a situation where we have a microorganism. We need to treat you because you have an infection, but we don't want to destroy your cells in the process. So we need to know what is something that is specific to amoebas or to bacteria. So we can target those differences and we can kill them whenever we need to kill them, whenever our white blood cells can't do the job. Okay. All right. So let's kind of talk about some things that we can target here. So I'm going to switch gears here. I'm going to talk about fungi because fungi really hard to kill. Like if you've ever had a fungal infection, like you had a fungal infection in a toenail, in a fingernail, ringworm is a fungal infection. Those things can be really hard to kill because fungal cells, there's not a lot of treatments for them because fungal cells are really similar to our cells. Because fungi, plants, and animals are all eukaryotic cells. So since they're all eukaryotic cells, they have a lot of parts in common. And so it's hard to find a difference between fungal cells and our cells. So when we give you a medicine, it doesn't damage your own cells. As a matter of fact, if you do develop a toenail fungal infection and you decide to take there's internal medicine you can take now that's supposed to clear up your fingernails or your toenails of that infection, but it also can cause liver damage to some individuals. So again, we need to figure out ways that we can exploit cells and kill them and not kill our own. So with fungi, here's one thing that we can exploit is they have cell walls. We don't. And if you remember, we talked about their cell walls are made of a carbohydrate, which is called chitin. So if we can design a molecule that will destroy the chitin, then it can destroy the fungal cells, but not destroy our cells. Okay. So again, some of these little features and these very specific pieces of information that we're putting in here, you're going to be like, why do we have to know this? Well, again, it's because if they have unique features, we can possibly target those features. They're kind of like a point of weakness here, okay? Speaking of cell walls and targeting cell walls, that's one thing that we can do with bacteria cells because they have cell walls and their cell walls are made of a different carbohydrate. It's called peptidoglycan. And I knew I was going to talk about it in this unit, peptidoglycan, which is why I kind of introed it to you in the biochemistry unit. Here's one way to help you remember peptidoglycan, right? Let's say you ate some bad food. You got some bacteria and it's causing you to have a terrible stomach ache. What's the pink stuff that you can take that will soothe your stomach? Pepto-Bismol, right? So you might now be able to relate peptidoglycan. It kind of sounds like Pepto-Bismol. Imagine bacteria giving you a stomach ache. Okay, so link peptidoglycan to bacteria cells. Okay. All right. Now, one thing in addition in lecture 1 that you need to get out of lecture 1 is that there's this idea of common ancestry. Now, we know that this is true, that if you and someone else have a common ancestor, you guys are going to have traits in common because those traits are getting handed down by genetics through DNA, right? So sometimes you guys are going to see, and they're going to seem maybe like complex questions, complex essay questions. It says, compare the following organisms to identify shared traits due to common ancestry, okay? This is just a fancy way that says list similarities. If you list similarities between you and your cousin, okay, you're listing evidence that you have a common ancestor, okay? So if it talks about indicate evidence of common ancestry, you just need to list what are some things that are similar because they had a common ancestor. So let's talk about bacteria and protists. Now, from the lecture, you would have to get and be able to understand that protists are eukaryotic cells. So really, this whole table, you could know nothing about protists. But if you could identify at least that they're eukaryotic cells, filling out this table is like saying, what are some similarities between prokaryotic cells and eukaryotic cells? And what are some differences? Okay, what are similarities and what are differences between prokaryotic cells and eukaryotic cells? Okay, so just because I throw protists in there, I could put Giardia lamblia in this spot right here. And if you have no clue what it is, but the text says, oh, it's a protist, then you have to be like, oh, it's a eukaryotic cell, okay? So first of all, let's talk about similarities. Similarities indicate common ancestry and relatedness, okay? So first of all, every single organism on this planet is made of DNA. The DNA is made of the same 4 bases. So the same 4 bases code for the proteins in all living organisms. That's because there's a common ancestor. Another similarity is that if DNA codes for proteins, then you need those ribosomes to build those proteins. So all organisms have ribosomes. Now, we can actually exploit some differences between ribosomes. So there's some medicines that we can take that can destroy ribosomes of bacteria cells because they are slightly different. They're smaller, and there's some changes in the structure molecularly of bacteria ribosomes, okay? They have smaller ribosomes. You'll learn that ribosomes are made of ribosomal RNA, okay? So I'm just kind of putting that there. And their ribosomal RNA looks different from our ribosomal RNA. So we have medicines that can target and destroy their ribosomes. And then we're going to learn more about cell parts, but hopefully a lot of you already kind of remember things about what ribosomes make those proteins. And if you destroy their ribosomes, they can't make proteins, and they're not going to be able then to make enzymes and other things to assist in reactions, and they would die, okay? One thing you're going to notice a lot on this table is these 4 things are all eukaryotic. So these 4 parts of that table are going to all look the same, okay? Because these are very closely related. They're all eukaryotic cells. They're more closely related to each other than they are to bacteria cells. So they're going to have more things in common than what they do with bacteria cells because they're simply more related, just like you and a cousin or a brother or a sister, okay? So let's go back up here. All organisms have DNA, right? Well, if you remember, prokaryotic cells, they have DNA, but it's not in a nucleus. No nucleus. Okay, so DNA is not protected, whereas in eukaryotic cells, they do have a nucleus. Now let's talk about that DNA. The DNA that we see in those bacteria cells is circular. There's no beginning and end to a bacteria cell chromosome. DNA just looks like a circle. Whereas in all eukaryotic cells, they have what's called linear DNA. So there's going to be a beginning and an end to every single chromosome. Okay. Another huge difference is those bacteria cells are very tiny and they're very simple. They lack membrane-bound organelles to help them do a lot of unique tasks that eukaryotic cells can do because they do have those organelles. So since protists are eukaryotic cells, that means even though you know nothing about protists for the most part, you do know, if you know anything about eukaryotic cells, that they have membrane-bound organelles. So we're talking about membrane-bound organelles. Talking about Golgi and Smoothie R and Roughy R, okay, and all those parts that we draw and we label in cells from past classes. Even when you're younger, you guys were labeling organelles and stuff like that, okay? All right. Now, let's talk about the advantage, what organelles offer for eukaryotic cells. So I'm going to flip over and go to number 7, okay? So one thing that you have to know in this unit is you have to know similarities and differences between different types of organisms. Now, to simplify things, just remember, everything on this planet is eukaryotic, except one thing, bacteria. They're their own class. They're prokaryotic. Everything else is eukaryotic. So if I say a octopus, prokaryotic or eukaryotic? Eukaryotic. If I say hydra, you're like, what the heck's a hydra? Well, it's not a bacteria cell, so that means it's eukaryotic, okay? So really simplify it. almost everything on this planet is eukaryotic, except for those simple little bacteria cells. All right, so speaking of the fact that we have organelles and they don't, let's talk about the advantages of organelles. And this whole slide kind of sums that up for you. It's the fact that we have organelles gives us more surface area for reactions to take place. Okay, so one advantage is organelles give more surface area. I'm going to put SA for reactions to take place. Okay, and notice that a lot of their membranes are folded. So I'm going to show you kind of two things here, this versus something else. So this is my rough ER. And rough ER, we're going to learn, is rough looking because it's covered in ribosomes. That's what these little dots are, the ribosomes. Okay. So if I space out these ribosomes, okay, I end up getting 4 on that rough ER. Now I'm going to draw a rough ER that is the exact same size and would take up the same space in a cell as this structure. But this rough ER is folded. Let's look at how many ribosomes we could pack, and I'm going to space them out the same. I'm going to give them the same space on this membrane between them, and look at how many ribosomes we can pack in that area now so that more reactions can take place in that same area, okay? So let's kind of indicate, remember, this folded thing is an important thing. It's going to come up in many units. If you fold something, it increases surface area where action can happen. And more surface area, more action, usually the more a cell or an organism can do, or even a cell part can do, or even a tissue can do, and organs can do if they have more surface area, okay? Another advantage goes along with the mitochondria. So basic mitochondria fact is it makes ATP, right? And it's actually a really complex process. I think there's like 30 some steps to make an ATP molecule. And we're gonna talk about those in unit 4. That's gonna be an exciting unit, I'm telling you. But it really just boils down to this. The reason why mitochondria can make ATP is because they can do this. They can put a ton of protons. So remember, H pluses are protons. They can gather and concentrate H plus in this space because they have membranes. So they have an outside membrane and an inside membrane, so it creates a space. And because they can put a bunch of protons in that space, they can use those protons to make ATP. So this is kind of showing you some of those other advantages is organelles can enclose and they can isolate molecules for reactions. Okay, so they can enclose and isolate molecules so that reactions can take place. Okay. Now, as for the isolation, let's add a little something to that isolation. Let's talk about a cell part, the lysosome. And if you remember, lysosome is the cleanup crew of the cell. They contain these digestive enzymes that can break down molecules. Okay. The problem is those digestive enzymes break down proteins, carbs, lipids, and nucleic acids. Well, that's what your entire cell is made out of. Proteins, carbs, lipids, and nucleic acids. So you better hope those suckers don't get out, okay? So the fact that they keep these digestive enzymes away from other molecules in your cells is a good thing. Otherwise, lysosomes would digest your own cells. And as a matter of fact, when your cells get a signal to die, and that's normal, cells get signals to die, it's called apoptosis. The lysosomes will release their digestive enzymes in that cell and will break that cell down, the proteins, the carbs, the lipids, nucleic acids, okay? All right, so how did all of those cell parts get inside of a cell? So a little bit of evolution in this unit. So we're going to talk about this theory. It's called the Endosymbiotic Theory. And you have to understand that if something is a theory, that means there is considerable evidence that supports that idea. As a matter of fact, no evidence has been found to contradict this idea. And that's why it's made it to this level of a theory, because there's just lots of evidence that supports it. So let's talk a little bit about what I'm showing you here on the screen. If I asked you to name the cell part, like if I made this a much smaller diagram and I point and I say, name that cell part, what might you name this cell part right here? Thinking back to cell parts and labeling them. Does anyone kind of recognize this part right here, what we maybe would name it?
Is that the Golgi?
Right, it looks similar to a Golgi. It also looks similar to the Refiar, right? And so now let's blow this thing up and look at really what's happening here. So this is the plasma membrane, and you guys learned in the biochemistry unit that the cell membrane is made of a phospholipid bilayer, right? Well, we know this happens in cells, is membranes can fold inward. They can fold inward into the cytoplasm. And if this area of the membrane and this area of the membrane meet, now you have a compartment on its own in the cytoplasm. And this compartment can contain molecules where reactions, and they can react with each other. So now we can have an isolated area in the cell where reactions can take place, okay? Now, let's see if this makes sense. If this is all made of the same thing, right? If this is all a phospholipid bilayer here, then these cell parts that we have in our eukaryotic cells should also all be made of a phospholipid bilayer, and they are. So this is how we believe that certain cell parts were made in cells. Let me show you this quick animation, okay? And like I said, this even happens in bacteria today. Prokaryotic cells. We can look at them in a microscope and we can see infoldings of their cell membrane. So this isn't like out of the blue that something like this just happened, and it only happened like early, 3.5 billion years ago. It still happens today in bacteria cells. So even bacteria cells, sometimes they look like they have little organelles. Here's some infoldings right there, okay, that look just like the cell membrane, okay? So that's how we got certain parts of cells, but let's talk about two very different cell parts. They originated a different way. So we believe that the ancestors to mitochondria and chloroplasts were once free-living prokaryotic cells that got engulfed by another cell and just didn't get digested, okay? Now here's what I mean by that. Let me go ahead and I'm going to play this for you here. So this is one of your white blood cells. I love this. They're showing you a 3D model of white blood cells. Now, a 2D model looks like a blob, right? Here's a white blood cell, and I love this video because it's kind of crazy to think that white blood cells can do this for us. But this white blood cell is just chasing a bacteria cell, and it's going to engulf it. It's going to wrap its membrane around it and bring it in. And for a split second there, we have a cell. Go back here. Come on, computer. We have a cell inside of another cell, right? Well, if this white blood cell didn't digest that bacteria cell, then that bacteria cell kind of just found a new place to live. And that's what happened with these very early, very simple cells, is that one engulfed the other and didn't digest it. Now, there must be evidence to support this, right? So let's go back here. So if this is a bacteria cell, what does that cell look like? What are the structure or the features of a bacteria cell? Well, here I have a bacteria cell. It has its own ribosomes. It has its own DNA. The DNA is circular. Think about the size of a bacteria cell, right? They're much smaller than eukaryotic cells. And so when we take a look at mitochondria and chloroplast, we actually see the exact same thing. I really wish that I could, let me try this real quick here. See if this is going to work for me. So again, to use that Paint on tab tool, you have to have something opened up and it has to be in Google. So let's see if I can open this with, come on, open for me. Okay, so if I open my PowerPoint or my worksheet or whatever, and I put it in, I open with Google, yay, now I can draw on it. So I can kind of show you this a little bit better here, okay? So imagine we have two simple cells here, okay? And this cell wraps around and brings this cell inside of it, okay? This bacteria cell that got engulfed still has its features. It has its own DNA. It would have its own ribosomes. It should be able to grow and divide on its own because it could do that when it was outside the cell. And what I'm listing is all features of mitochondria. Mitochondria have their own DNA. They have their own ribosomes. If mitochondria, they multiply on their own inside of cells. So this is one cell and it's showing you the mitochondria inside of it. Look how they're kind of like moving around like on their own, like little worms in the cytoplasm of this cell. And notice there's some long ones and then there's some short ones. The long ones are about ready to divide. And this is what bacteria cells look like whenever they go to divide. They elongate, they copy their DNA, and they split in two, okay? So you're going to see questions where you have to indicate evidence of this theory, the endosymbiotic theory, and that kind of goes along with this question right here. So on number 6, it says, diagram or label 4 different features that offer evidence that the ancestor to this organelle, this is mitochondria, its ancestor was a free-living prokaryotic cell, okay? So here's some evidence. First of all, they have their own ribosomes. They can make their own proteins. They do not need proteins made by the ribosomes in the cytoplasm of the cell that they're in. They can make their own proteins. Now, to make their own proteins, they need their own DNA. So they have their own circular pieces of DNA inside of them. And by the way, when you look at the genes on mitochondrial DNA, we see very, very similar genes on bacteria cells today. So they have similar genes. And just like you and a cousin, you have similar genes. Why? Because you have a common ancestor, because you're closely related. Okay, now I'm going to show you this diagram so that this next part makes sense here. Actually, let's go back to, which one do I want? Try this one. So notice that if this is now inside of a cell, this cell has 2 membranes. It has the membrane of the host because the host cell has to wrap its membrane around a cell to bring it in, but then it has its own original membrane. And that's something that we see in mitochondria and chloroplasts, 2 outer membranes. So that's why they're the only 2 structures in cells that have 2 membranes. So that's why it tells us that they originated differently than these other cell parts, like the rough ER and the smooth ER and so on. Also, this is going to elongate, copy its DNA. It's going to divide. So cells just simply get more mitochondria and more chloroplasts by them growing and dividing inside of cells, okay? One thing I do want you to know, though, is that mitochondria and chloroplasts are dependent on their host cells. They cannot live on their own as individual cells. So with mitochondria and chloroplasts, they rely on the cell to make certain proteins to actually kind of keep the mitochondria and the chloroplast intact so they don't degrade and break down over time, okay? All right, with that being said, we're going to go to our breakout rooms for the next 15 minutes, and I'm going to have you guys work together to finish up and answer as many parts as you can to this assignment, okay? Remember that if you have to retest anything, you have to have these worksheets all the way done. So the more that you do now together, where there's multiple brains coming up with answers, the less you will have to do if you have to retest. Otherwise, it's going to end up being quite a bit of work, okay? All right, so let me go into chat here and just repaste your groups and your links. And then I'm going to go ahead and start opening those breakout rooms. When you guys are in your breakout rooms, I expect you to have your cameras on so I know you're there. It would be nice if one person could share their screen. And in Google Classroom, you guys have this. You can open it up in Google Docs and draw on it and stuff like that if you need to. And like I said, more brains are better than one. So work together to get that sucker finished, and I'll be in and out of your rooms.
Miss Spencer?
Yes.
The list you sent is from your first walk, by the way.
Thank you very much. Let me go back and zoom further down. I guess I didn't realize that was that far up. There you are. Try that again. All right, there you go.